Hello everyone, in this lecture we'll talk about the oxidative phosphorylation, which is a process that will convert FADH2 and NADH into even more ATPs, and it happens right in the inner membrane of the mitochondria. Well, everybody cannot survive without oxygen, but why do we need oxygen in the first place? Well, we need oxygen in this reaction, which is the oxidative phosphorylation. From the name, it tells you that it is an oxidation process which requires the presence of oxygen, and it is also a phosphorylation process, which means the addition of a phosphate group. So what is being phosphorylated over here? Yes, the ADP is being phosphorylated to become the ATP, which is the energy currency for the cell. So remember, in glycolysis, we only produce 2 ATP per glucose molecule, while in the Krebs cycle, we have 2 GDPs for every glucose molecule. And we do have a lot of NADH and FADH2, which will then be harnessed in this process to produce even more ATPs. So the ultimate purpose of this reaction, oxidative phosphorylation, is to release the energy from the electron carriers, NADH and FADH2, to produce many more ATPs. And this happens right in the mitochondria. To be very specific, again, it is in the inner membrane of the mitochondria in a structure known as the electron transport chain. Okay, these are the learning outcomes for this session. First of all, to understand the importance of oxygen to the aerobic organisms. Number two, to name all the components of the electron transport chain. Number three, to describe how electron movement can establish a pH gradient, which is then used by the ATPase to generate ATP molecules. Okay, here's a very quick recap. At the end of the Krebs cycle, we are left with these high energy molecules, which are NADH and FADH2. So they are high in energy because they have captured the high energy electrons from the sugar molecules. But these molecules are not the universal energy currency in the cell, which is the ATP. In other words, we have to convert these molecules into the ATP in the final process of metabolism, which is the oxidative phosphorylation. As mentioned earlier, in the oxidative phosphorylation, oxygen would be used to further oxidize these high energy electron carriers, which are NADH and FADH2, so that the electrons can be released and that energy stored in the electrons can be used to generate more ATPs, while oxygen itself is being fully reduced into the water molecule. And this reaction will happen in the structure known as the electron transport chain that is found right in the inner membrane of the mitochondria. So for information, this reaction itself is a stepwise process, which means it happens according to a specific sequence which involves multiple steps. So in the electron transport chain, you have three important electron carriers, which are coenzyme Q, cytochrome C, and the oxygen molecule, which is the final electron carrier in the series. And you also have four protein complexes, complex 1, complex 2, 3, and 4. So for complex 1, it is NADHQ oxidoreductase, which is responsible to receive the electron from NADH, while complex 2 is succinate Q reductase that will receive electron from FADH2, and the role of complex 3 Q cytochrome C oxidoreductase is to shuttle the electron from coenzyme Q to cytochrome C, and the role of complex 4 cytochrome C oxidase is to shuttle electrons from cytochrome C to the final electron carrier, which is oxygen molecule. So the problem is, how do we make sure that the electrons will follow the right sequence? Well, it all depends on the electron transfer potential of each component, or it is also known as the redox potential for each component. When a component is having a negative redox potential, it means it has a higher tendency to donate its electron as compared to a component with a positive or with a higher redox potential, which means the tendency to keep the electron. And therefore, electron will always flow from a component of a negative redox potential to the components with a higher redox potential. And here's one important thing to take note, that all protein complexes except protein complex 2 can use the energy from the flowing electrons to pump protons across the inner membrane into the intermembrane space. So it is the gradient of the protons built up in the intermembrane space that can be used to make ATP eventually. Now, let me just show you the simulation over here. So with the flowing electrons, we can capture that energy to pump protons across the protein complexes to the intermembrane space. And you can see that it only happens with complex 1, complex 3, and complex 4, but it doesn't happen with complex 2. And for this reason, NADH can generate more ATP than FADH2. So generally speaking, one NADH can generate up to 3 ATPs, while one FADH2 can generate only up to 2 ATP. Here you have the working principle of the electron transport chain, which follows the first law of thermodynamics, and it mentions that energy cannot be created nor destroyed, but it can be transferred from one form to another. So to generate ATP, where is the source of energy coming from? Well, they're coming from the flowing electrons in the form of electron motive-force. So protein complex 1, 3, and 4 can utilize this EMF to pump protons across the inner membrane into the intermembrane space. So with that, this generates a gradient of protons concentration, which is known as the proton motive force. And as the protons rush back to the matrix by flowing through this ATP synthase molecule, the energy is converted into the chemical energy, which is in the form of ATP molecule. Just in case if you are not familiar with the structure of mitochondria, they are double membrane organelle, and the space in between the outer and inner membrane is known as the intermembrane space. And in the center, you have liquid known as matrix, which contains all sorts of enzymes and substrates for metabolism. And to increase the total surface area, the inner membrane in blue will fall into cristae, which is as big as 3 football fields. So what you need to remember is that glycolysis happens outside of the mitochondria because they take place in cytoplasm, while citric acid cycle happens in the matrix, while electron transport chain is found in the inner membrane of the mitochondria. In this diagram, you have the change of free energy across the electron transport chain. So it begins with NADH, which donates a pair of electrons to complex number 1. And with each transfer of electrons across the series, you have the drop in free energy. In other words, delta G is equal to negative values. So over here you have an exergonic reaction. Now if you can remember, the hydrolysis of ATP releases about 30 kilojoule per mole of energy. So over here you have roughly negative 150 kilojoule per mole of energy released from one NADH. So my question is, how many ATPs do you think one NADH can make? Well, the answer is not 5, but it is 2.5 ATPs. Why is it so? That's because not all energy are used to make ATPs, and you also use part of the energy to combine the hydrogen with the oxygen to make water molecule, which is part of the side product in this reaction. And a second reason is that you have the loss of energy, because according to the law of thermodynamics, the conversion of energy is always accompanied with an increase in entropy, or delta S, which is the unusable energy within the system. Now, a second thing that I want you to focus is the presence of iron in all these protein complexes 1, 2, 3, and 4, and also the cytochrome C. So we know that iron is a transition metal, so it has several oxidation number. And the function of the ion over here is to receive and donate electrons across the series. When it receives an electron, it becomes less positive, so it becomes a Fe2 positive ion. When it donates an electron, it becomes more positive, so it becomes a Fe3 positive ions. And the next thing I want you to look at is ubiquinone, which is also known as coenzyme Q. Well, it is not exactly a protein, but a small hydrophobic molecule that shuttles in between complex 1, complex 2, and complex 3. And one thing you have to remember is that all these protein complexes are embedded in the inner membrane of mitochondria. They are less mobile, but the small ubiquinone and cytochrome C can move around in the inner membrane, and they are transferring electron in a much efficient manner. Over here, I just want to show you how these protein complexes in the electron transport chain are embedded in the inner membrane of the mitochondria. And to be more accurate, protein complex 1, 3, and 4, this will span across the whole inner membrane, while for complex 2, ubiquinone, and cytochrome C, they are just peripheral molecules that will not span across the whole membrane. And this is what happens in the oxidative phosphorylation. When electrons are transferred from one component to another, the release of the free energy is used to change the conformation of the protein complex. And as a result, they pump protons across the inner membrane into the intermembrane space. So this happens for complex 1, complex 3, and complex 4, while it doesn't happen for complex 2, because protein complex 2 is not a transmembrane protein, which means it cannot form a channel to transport protons across the membrane. And there's one more thing I want you to see, which is the amount of proton pumped by each protein complex. You can see that both complex 1 and complex 4 pump a lot more protons than complex 2 or complex 3. Why do you think so? Well, just look at the change of free energy. Because with protein complex 3, it has the smallest change in free energy, and therefore it has the smallest energy, and with that you have fewer protons pumped across the protein complex. Eventually, this forms up a high proton gradient in the intermembrane space, which is in the form of proton multiforce, which can then be used to generate ATP. Okay, now we will look at a bit more details and what's happening in each process. So the oxidative phosphorylation begins when NADH transfer their electrons to the first complex. At the same time, NADH also releases one proton. So it has now become NAD positive, which can be reused in the Krebs cycle. So for information, complex 1 is a very huge molecule. So the first functional group to receive the electrons from NADH is a prostatic group that is attached to complex 1, known as FMN, or the flavin mononucleotide molecule. So in doing so, FMN is being reduced to FMNH2, and then the electrons will be transferred from FMNH2 to a series of iron-sulfur cluster, which is another prostatic group found in the complex 1. Okay, an iron-sulfur cluster has a central iron that is coordinated by 4 cysteine side chains, and you can find similar iron-sulfur cluster in other protein complexes as well. And finally, the electrons are transferred from the FMNH2 or from the FES cluster to ubiquinone, which is coenzyme Q. So with the transfer of electrons in this process, the energy release is used to pump 4 protons from the matrix into the intermembrane space, while another 2 protons are now bound with coenzyme Q, forming QH2. So overall, 4 protons are pumped into the intermembrane space, while 6 protons are removed from the matrix. So this forms an initial gradient of protons. Okay, now the electrons is with the coenzyme Q, which is also known as ubiquinone, because it is ubiquitous or commonly found in many species. And again, as I mentioned, ubiquinone is a hydrophobic molecule that allows it to diffuse very rapidly in the inner membrane of the mitochondria. Now, here's the thing. For ubiquinone to receive the electron from FADH2, it must first go through complex number 2. Okay, so FADH2 will first donate electrons to complex number 2 before it can be transferred to ubiquinone. And as I mentioned earlier, complex 2 doesn't pump any proton across the membrane, and for this reason, FADH2 will generate less ATP than the NADH, which will pump from complex 1 that pumps protons, subsequently followed by complex 3 and 4. Then electrons are passed from ubiquinone to complex number 3, or the cytochrome C reductase complex. The transfer of electrons from ubiquinone to complex number 3 is known as the Q cycle. So why is it a cycle? Because it is a cycle that allows the regeneration of the oxidized form of the coenzyme Q. So from QH2 to Q, that is oxidation, while reduction happens with your cytochrome C reductase, which means the complex tree itself is being reduced in this reaction. So in this process, two chemical protons are removed from the matrix to reduce complex number 3, while 4 protons, they are pumped into the intermembrane space. So with that, you have got a larger difference or a larger potential in terms of the proton gradient. But don't worry too much about the numbers. It's important that you understand the process. Okay, next, electrons from the complex tree will then be passed to cytochrome C, which is a small soluble protein. And this cytochrome C can only take 1 electron each time. As a result, you will need 2 of these proteins for each transfer. Now we know that cytochrome C is a soluble protein, okay? So do you think it could be a transmembrane protein that is embedded within the lipid bilayer? Well, the answer is no, because if cytochrome C is soluble, it means it must be hydrophilic, and as a result, it is located at the periphery of the inner membrane, which is found on top of the surface of the lipid bilayer, rather than be embedded within the structure of the lipid bilayer. Okay, here comes the final complex, the cytochrome C complex. They will receive electrons from cytochrome C and pass them to the final electron carrier, that is oxygen molecule. So in this process, 4 electrons from the cytochrome C will be transferred to oxygen, and at the same time, 4 protons are transferred to the oxygen so that it will be reduced to form water molecule. And due to the conformational changes with complex 4, 4 more protons are pumped across the membrane into the intermembrane space. To summarize, 4 protons are pumped from the matrix into the intermembrane space, and 4 more chemical protons are removed from the matrix to reduce the oxygen into water molecule. So at the end of the electron transport chain, we have built up a gradient of 12 protons in the intermembrane space, and we have removed 16 protons from the matrix. So there is a great difference in terms of the proton concentration as well as in charges. So remember, protons, they are positively charged. So this forms a great potential. So let's come back to the first question. Why do we need oxygen? Well, I think now you know the answer. Because oxygen is the final electron acceptor, and without oxygen, electron will get stuck in complex 4, and eventually they will clog the whole electron transport chain.